So yeah, I'll be focusing on ecological network structure research, and this is necessarily both very partial and very biased view, but I hope to touch on a bunch of different sort of things coming down the pike and that are just starting to happen in this kind of research. And I'll be focusing in particular on food webs, trophic interactions between organisms, like shown here, for a lake ecosystem with algae and zooplankton, fishes, and even some parasites. And while this is just one example of a type of interaction between species, feeding interactions are really central both to ecological and evolutionary dynamics, so they're a good place to start. I believe Stefano will probably be talking about a wider array of interactions in his talk after mine. So first to focus on data, generation zero. What was generation zero? In 1880, Lorenzo Camerano, Italian scientist, published the first known graphics of ecological networks, of feeding networks. And so up here on the left is a generalized trophic network with 5 different categories. But then he also did a specific trophic network over here on the right that included 15 different types of taxa. And so this was published more than 100 years ago and then was promptly lost to science until about, 20 years ago when Joel Cohen found it and had it translated and published it and commented on it. But just to show that the roots of network research and ecological network research actually go back further than you may even think. So after generation zero, we pop up to the 1970s and 80s to what I would think of as generation one data. This is a bunch of data sets, mostly drawn from the literature. Joel Cohen and his colleagues compiled them into a database called EcoWeb. So these food webs had about 2 to 90 species, but on average they only included 20 species. And this is up here is an example of one of those food webs that came out of Charles Elton's work in the 20s. So these are very small representations of the diversity of a system. Generation 2 data, if we jump up to the 1990s basically, and 2000s, now we basically have jumped up into a higher level of diversity being represented in ecosystems. And now we have a set of about 20 to 30 food webs that we tend to work with about 25 to 200 taxa, a mean of about 70. This food web in particular is the Little Rock Lake food web that Neo Martinez published in 1991. And so you have a higher level of resolution, and these are the data sets that many of us and several of us in this room have kind of pounded on for the last 10 years and done a lot of interesting analyses and modeling, which is great, but these are still really, Little Rock Lake, it's not really true. This is a highly resolved data set, but a lot of these data sets we're working with are still fairly impoverished versions of the species diversity in systems. So, but on the basis of these data, some of both generation 1 and 2 data, a number of key questions have been asked, things like how are food webs organized? Are properties of food webs scale dependent? Do food webs from different habitats have similar structure? Are there simple models that predict that structure? How does food web structure influence robustness or the risk of cascading extinctions? And does structure influence the dynamical persistence of species? And these are all really important questions that I think we've made a lot of progress on based on these data. However, now I think we're moving into an era of what I refer to or think of as generation 3 data. And there's a lot of different characteristics. Food web research, as with ecological or with network research in general, has really exploded over the last decade and is going in a lot of different directions, which is incredibly exciting. And so in terms of the empirical side of things, I think some of the things that we're starting to see is much more comprehensive data that's much more highly diverse, and that's starting to represent better the true diversity of systems. And also that's more resolved at all different levels, so that by resolution I mean things are being resolved to the species. In the older data sets you often see invertebrates and plants all grouped together, and the vertebrates, the things that people like to study, are all very highly resolved, so we're trying to get beyond that. We're augmenting our data with a lot of additional information, trait information like body size, flow information for the links, so phylogenetic information about evolutionary relationships. So we're basically annotating both our nodes and our links with more quantified information. We're also doing a lot more with time and space, spatially explicit data, looking at basically data that goes across spatial scales or across space or through time, through deep time. An exciting thing that's happening is people are putting together large databases of replicated food webs. So instead of just these singular food webs drawn from different systems, we're now getting people who are putting together hundreds of food webs for a particular kind of system and doing that along a gradient, like a climate gradient or a spatial gradient. We're getting more work on experimental food web research, and connecting back to what Ian was talking about. We're also moving towards some work on individual-based data, and which I think is going to be really important. There's work on interdependent networks, multiple types of interactions between the same species within a system. And then also just on the tech side, we're still really data limited. It's hard to put together really good data. And so there's various people working on aspects of automated data generation, both sort of in the field sense, but then also trying to develop semantic web technologies in order to synthesize data from electronic databases and bring it together. So I'm just going to give a couple of examples of things that I'm involved with that I think reflect a couple of these trends, and people are working on different aspects of them. My first example is highly resolved paleo food webs. And so here we have the geologic timescale. Here we are up in the Holocene. And here's the whole Phanerozoic, the era of multicellular life on Earth. And I want to talk in particular for a moment about the Messel Shale, which is in the Eocene, 49 million years ago. And this is a picture, an artist's rendering of one of the links in this food web, which is basically this 8 to 9 foot tall bird that has the common name of terror bird, I think for fairly obvious reasons. It was a major predator in the system, and it's a little hard to see, but in its mouth is a terrier-sized horse. It's one of the early horses. So this is actually a link in the food web of the Messel Shale. So when we go back in time, I mean, how is it possible that you can get feeding information for a large number of species in an ecosystem that's very ancient? In a lot of cases, you can't. Most fossil assemblages just preserve the hard body bits. But in some cases, like with the Messel Shale, you get excellent preservation of soft body parts. And there's a lot of ecological evidence within the fossil record, especially for the Messel Shale. So this shows examples of some of the taxa and links in the system. One of the best lines of evidence that we have is gut contents. And there can often be quite a bit of it. Here's gut contents for one of the little horses, gut contents for a hedgehog. There's also a lot of evidence from insect damage on plants and also the functional morphology of different species. And I've been collaborating on this project with Conrad Labandera of the Smithsonian Institution. He's the world's expert on fossil plant-insect interactions. So he can actually go and identify a plant leaf to the plant species and then look at the patterns of damage on that plant and trace that back to the particular mouth parts of a particular insect. So it's actually really remarkable. So when you put it all together, this is what you come up with. This is a food web for the Messel Shale. It has 700 taxa, over 6,000 feeding links. And so of the taxa, there's a huge array of types of organisms represented. The orange shows vertebrates. The yellow shows invertebrates. Blue shows bacteria, fungi, detritus, and the green are plants, algae, and diatoms. And one of the things about this data set is it's the first food web we have for any ecosystem, including extant ecosystems, where we have highly resolved plant-insect interactions for a terrestrial system. I mean, to me, that's kind of crazy that we've gone back in time 49 million years and have come up with a better data set than we have for extant systems. And so I'm hoping to sort of inspire terrestrial food web people to put together better data for extant systems. And just to show, we can also split this full food web into a lake food web and a forest food web, and start to do a lot of different kinds of analyses on these data. So my second example is basically looking at more comprehensive coverage of the types of species and ecosystems. Most food webs that have been compiled so far have really focused on free-living species. And of course, a huge amount of the diversity of systems in the world is parasites. And food web people have pretty much, or ecologists in general, have mostly ignored parasites. Parasitology and ecology have kind of grown up apart from each other, but they're starting to come back together. And so there's a working group that brought together parasitologists and food web people that's put together a bunch of really amazing new data sets with highly resolved parasite information included. So here's your food web without parasites. So this is and this is for an intertidal system, about 91 taxa, 770 links. When you throw the parasites in there, they represent a third of the species in that system. And that's not even covering all the possible parasites in this system. So clearly we're missing something by not including parasites. And 75% of the links involve parasites. So again, this is a situation where we can drastically improve the quality of the data and the coverage and comprehensiveness of the data and ask new types of questions. And then my last example on the data side are these kind of multi-web databases that are coming online. And this is just one example of a project I'm involved with in the Aleutian Islands with intertidal food webs, where my colleagues have put together data at different spatial scales. And so we have over 700 food webs at 4 different spatial scales, at the meter squared scale, and then the transect scale where you aggregate across the quadrats, and then the site level, and then the island level. And one of the questions that we just haven't been able to answer really is, how does spatial scaling impact our understanding of ecosystem organization? So we've got data 3.0 coming online, and that opens up a whole huge world of new questions, of questions 3.0. Some of those include things like the scale dependence of structure, and this has been looked at previously in different ways, and Neo Martinez did a lot of really important work on this. But this opens up new ways, and we can extend our understanding of scale dependence as we get these these databases and these more highly resolved data. Community structure, something that Luis talked about, and in this case, I would say a big challenge is to move beyond, he's done from moving beyond the algorithm to validation. I want to move beyond validation to novel insight. And some studies are starting to get at some of that, and Stefano's done some nice block modeling, group modeling for food webs. But I think it's really important that we figure out that it doesn't just reify what we already know about a system, that we actually can come up with new hypotheses about what's going on in communities. As you add different taxa in, are they playing different roles in the system, and does it lead to different kinds of organization in the system? Are parasites, do they play different topological roles in other kinds of predators? That's one of the things we're looking at. We can start to ask interesting questions about macroevolution with the deep time data. We can do things like do more to explain variability in structure in terms of traits and flows and other kinds of information. I also think another thing is we really need to develop a theory of trophic organization. We aren't really there yet. We're still kind of phenomenological. And so, are the kinds of patterns we're seeing, are they different from a neutral or a null model? Do they maybe have something to do with thermodynamics? Basically, there's just a lot of theory development I think that can go on. And another really critical thing I think will be in the future is to go from the individual level up to the ecosystem level. A lot of food web work right now, most of it focuses at the population species level. And really, we'd like to know, as you go from from individual interactions and traits, how does that ultimately impact ecosystem level function, basically passing through population level dynamics? So now I want to talk a little bit about analysis and modeling. So generation 1 in this case, and this goes along with generation 1 data, is the Cascade model, which was published by Joel Cohen and colleagues in the 1980s. And this is basically a very simple model of how species feeding interactions are organized. And it seemed to do a pretty good job of generating the structure that we see in these first generation data sets. But then we moved to the generation 2 model. And so this is the model, the niche model that was published by Rich Williams and Neo Martinez, both of whom are here. And this is one of the papers that Luis said made him jump up and down. It's a really, really nice paper published in Nature in 2000. It's a slightly more complicated version of the Cascade model, but it has some fundamentally different assumptions about organization. And it seems to do an amazingly good job of generating the structure that we see in these generation 2 data sets. And a lot of really cool work has been done by by people in this room, including Daniel Stouffer, paper in Ecology in 2005, and Stefano Alesina and colleagues in Science in 2008 that kind of builds on the niche model or explores the niche model. However, so as I've already said, with the 1.0 data and model, the Cascade model, and this early data, it ends up seeming or looking like we now know it's a bit of a house of cards. You have very simplified data and a very simplified model. They sort of reinforce each other, but they're not really reflecting what's going on out there in the ecosystems. So the house of cards came crashing down with a lot of Neo's work and other people's work. But then, of course, we have to ask the question, is the niche model a house of cards? And the generation 2 model and data a house of cards? As we move to the much more highly resolved and diverse and comprehensive data, what will the status of the niche model be? And we're just starting to be able to ask that question now. And I think one thing that's becoming clear is the fit of the niche model does decrease systematically as you go to larger and larger food webs. But it does so in a systematic way, and we're just starting to kind of pick this apart and understand it. And it may be that the niche model hits a really sweet spot with a particular size network and a level of resolution. So I would just want to mention a few sort of generation 3 approaches. There's a lot of stuff going on. These are just a couple of things. The first is kind of coming up with better null models, and I'll in particular mention maximum information entropy and talk about it just a little bit. There's also probabilistic maximum likelihood methods being applied to ecological network modeling and analysis. There's also people are starting to do individual-based models and analysis, again, kind of going along with the data issue that I brought up. And again, I think there's a lot of very interesting connections to some of the work that Ian is doing in his lab in terms of those nice predator-prey interactions that they're starting to look at. There's phylogenetic approaches, I think, that are very interesting, things that involve coalescent theory and just how you incorporate phylogeny and evolutionary relationships into analysis and modeling. And then also just in conversations with my colleague Chris Moore at the Santa Fe Institute, and also a lot of people here working on machine learning algorithms and how we can use those for looking at defining community structure, looking for structure and other aspects of network analysis. And that's just a couple of examples. There's many other things going on. So I'll just give 2 quick examples of those things. The first is, so this issue of degree distributions. This is, as Luis talked about, in food webs, we tend to see an exponential type distribution. And the work by, out of Luis's lab, Camacho et al., Physical Review Letters in 2002, a paper that I and Rich and Neo published in PNAS, and then work by Daniel Stouffer with Luis Amaral, basically shows this when you look at the raw data for food web degree distributions, there seems to be a lot of variation in the shape of the distribution, but then when you normalize it for links per species in the network, you find an underlying simplicity of pattern, a non-power law, more exponential type distribution. I mean, this is just something, this is an old result. Whoops. But the thing is, so like with the niche model and these successful models that people have worked on, they basically assume an exponential distribution. So there's not really kind of an explanation or a fundamental null model about the exponential distribution. And the principle of maximum information entropy provides one way to think about a null model. And this is work published by Rich Williams, who is here. And in this case, this is basically the issue that the probability distribution with the maximum information entropy is the least biased probability distribution, which satisfies a set of information containing constraints. And so you can use this approach very usefully as just to see if there's kind of a null expectation given certain kinds of things that you know about a system, in this case, a food web network. And so the constraints that we build, that Rich has built into this, is just the number of species and links in the system. And you can come up with tests for model fit. But again, the idea here is just sort of what is a null expectation? Because until we know what a null expectation might look like, we can't really start to look usefully for biological explanations. And this just shows, Rich and his colleague, Rich looked at 51 food webs, those basically generation 1 and 2 food webs. And this shows the cumulative distribution as a function of links per species. And so for this food web, for the St. Martin food web, for the resource distribution, the red line shows basically shows the empirical network and data, and the black line shows the central tendency or the prediction of the maxent model, and then 95% confidence intervals. And so in this case, there's a very good fit. There's no deviation of what you see in the empirical data and the maxent expectation. In these two food webs, consumer distribution and another consumer distribution, in this case, you see that the empirical distribution is wider than expected from the maxent, and here it's narrower. And overall, across these food webs, in about half the cases, you see no significant difference from a maxent expectation. So again, what this provides us is potentially one type of null model by which we, for those other 50% of the food web distributions, we can start to try to understand what's pushing things away from that null model, that maxent expectation. And if you're interested in maxent in ecology or maxent for other kinds of analyses, there's a book recently out in the Oxford series in Ecology Evolution by John Hart on the uses of maxent to look at macro ecological patterns. And it's a really good introduction. So my second example, also something developed by Rich Williams, is a likelihood-based approach. It's a probabilistic version of the niche model. And Stefano Alesina was the first person to basically use this type of approach for modeling food webs in his science paper in 2008. And Rich basically did an explicit version of the niche model using this type of approach. And in this case, what you're doing, it's a very data-driven approach, and you're using the data to come up with a maximum likelihood estimate of the parameter set for each species. And in this case, it's based on the niche model, so it's looking at those parameters, the niche position, the feeding position, and the feeding range for each species in the network. And then the PNM, the probabilistic niche model, assumes a non-zero probability of species I eating species J, depending on the mismatch between the niche position of the species being eaten and the feeding position of the species that's doing the eating. But the kinds of things you can start to look at, so in this case, it's actually looking at the overall structure and the detailed structure of the entire network. So you can actually see the fraction of specific links that are correctly predicted by this type of model. And then that allows you also to start comparing across some of these different models. And this graphic shows the fraction of links that are correctly predicted by 3 different models and across several different food webs. So the red crosses show a one-dimensional probabilistic niche model. The blue crosses show a two-dimensional probabilistic niche model. And these other circles show the results from a model published by Owen Pecci and his colleagues. And in this case, you can see that you're actually, the probabilistic niche model is doing a really good job of predicting over 80% of the links in most of these second-generation food webs. Adding a second dimension, it improves the fit slightly. And if you look at an Ikiki information criterion, there is an improvement with the second generation, or the second dimension added with this model. But you can also start to do some really interesting things by looking across the parameter sets. And then you can start to get back into some really interesting biological detail. And so what this graph shows is basically the prey for the whole set of predators for this food web. This is the Benguela marine food web. So the prey for each predator are ranked according to their niche value. And the predators are ranked according to the location of their feeding niche. And the black circles show the empirical links, so the links that actually are observed to occur between predator and prey. The gray circles within them show basically the probabilistic niche model's prediction of whether that link is going to occur. And you can see there's very good concordance between the observed and the predicted trophic links. But the other thing you can see is that there's very strong trophic niche contiguity in this data. And what I mean by that is that when you're lining up the prey, they are falling into a singular niche that's fairly contiguous. So you only get a few places where you break the contiguity. And this is a very fundamental assumption of the niche model and related models. And so this modeling approach provides a way for us to pick apart and to really look at some of those issues like intervallity in a more detailed way. And you can also impose body size on this and other kinds of annotated information. So for the last couple slides, I want to move on to the third part, which is application. And again, to echo some of Luis's comments, I think we need to really be thinking about how we use and apply these kind of new data and these new analysis approaches for things that matter, for problems that matter. And there's many problems that matter. Just the one that I'll mention here is the issue of humans and ecosystems and what their roles and impacts are. So I'm involved in a project in collaboration with archaeologists up in the Aleutian Islands. And so here's the tip of the Alaskan Peninsula, and here's the Sanak Archipelago where we're working. So here's a blown up picture of Sanak Island. And the questions that I'm interested in asking here and that we're interested in asking are what roles did pre-industrial humans, the Aleut, play in North Pacific food webs? How do human hunter-gatherers compare to other kinds of species in the system? And then what can we learn about sustainability from how pre-industrial humans interacted with other species? So we can bring a network perspective, an ecological network perspective, as a way, as a framework for trying to do some quantitative analysis and modeling of these questions. And so the first thing is to put together data again. What was the resource used by the Aleut and all the other species that they're interacting with and that are in the system? And by combining ecological observations along with basically information about Aleut resource use pulled from middens, from trash heaps, and then also ethnographic information and interviews, you can put together a really detailed picture of all the interactions of species in the system, including humans. This is just an example food chain from the food web for this system. For the intertidal food web, green algae at the bottom, shrimp, great sculpin, a type of fish, is eaten by stellar sea lion and then is eaten by Aleut. Of course, you can already start to fill additional arrows in here. The Aleut also ate the great sculpin, they also ate shrimp, and they also gathered and ate green algae. And so we can put this all together into, again, a very comprehensive and highly resolved, in this case, marine food web, the nearshore marine food web. And surprisingly, no one has ever compiled highly resolved data like this food web data that explicitly includes humans. And, a fairly obvious thing to do, but it just hasn't happened. And so in this system, we have 513 taxa that are trophically distinct. We have almost 7,000 feeding links and about 13 links per species. And again, I've color-coded the nodes here. Purple are birds, red are mammals, orange are fishes, yellow invertebrates, blue algae, and miscellaneous taxa. And the humans are back here in this hairball. So we can do just very simple things and just simple path length analysis. I mean, first of all, humans fed on 122 species in the nearshore marine system, so about a quarter of the species in the system. Okay, that's interesting, but how does it actually compare to other species? Here's just a simple histogram of the number of predators that eat a particular number of prey. So humans are way out here. They're basically number 2 in terms of their generality. Second only, very slightly, to Pacific cod, and then some other fish species down below, stellar sea lions. So humans were super, and we can do other things, humans were super generalist in this system. They're super omnivores. They're feeding at all trophic levels. They have the shortest, or near the shortest mean path lengths in this system. So they really are playing outstanding roles, topological roles in this system. And again, 96% of the species in the marine nearshore system here are within 2 links of humans. And, you know, this is fairly, I mean, people know that, you know, humans are having big impacts on marine systems. But this really quantifies how closely linked humans are to just about every other species in the system. So this is just a very useful tool for kind of really pointing out and showing the extent of the reach of human interactions. And we can take this type of topological information, you know, even this fairly simplistic type of analysis, and then we can use some of that in order to parameterize a nonlinear dynamical model based on bioenergetics. And we can use that in order to do some simulation experiments where we ask questions like, under what conditions can you invade a system with a species like a human that's a super generalist, super omnivore? And under what conditions will that tend to crash the system by resulting in a lot of extinctions of other species, and under what conditions will it tend to just kind of blend in to the dynamics of the system and allow for coexistence? And one of the things that we see in the system is that with the humans, or with a super generalist, a super omnivore, as long as they're only focusing on a few of their possible prey items at a time, it's very stabilizing for the whole system. And so one of the things that we know both about these hunter-gatherers and also about generalist predators is that they prey switch a lot. So they focus on a few things, and then when those become less available, they switch to something that's maybe a little less preferred but easier to get at. So they're constantly switching. And from our own work and from the work of other people, both in structure and dynamics, this is a very stabilizing dynamic for an ecosystem. And we can then start to compare this to the types of dynamics that go on in modern ecosystems or modern resource use. For example, the bluefin tuna fishery, where as people extract bluefin tuna from ecosystems, and it becomes harder and harder to catch and it becomes more rare, its value, its economic value skyrockets. And so instead of switching from bluefin tuna to some other resource, there's increased predation pressure on the bluefin tuna. And this is obviously bad for the bluefin tuna. I mean, everybody knows that. But I think what some of our work is starting to show is that that introduces a destabilizing dynamic for the entire ecosystem potentially. And so I think that's where you can start to really get at some really critical issues about what modern day humans are doing and how they're impacting modern ecosystems through resource management and resource use. And my time is up. There's a lot of collaborators that I can't go through on different aspects of projects I've mentioned, and many other people working on really, really cool things. And of course, acknowledging the funding. The program that we use to generate these types of visualizations, and you can actually do it's 3D visualization and dynamic animations. Neo and Rich and myself and others have developed, with Rich doing the bulk of the work. It's called Network 3D. If you're interested in it, just email me. And there's also a lot more information at foodwebs.org. And I thank you very much, and I look forward to talking to all of you offline, or some of you.